Good morning to all. I'll be talking on the very general topic that is introduction to microbial hazards. Several participants are from non-microbiological background, so it's a general kind of lecture. And this is a structure of my presentation that will be, there is a introductory part on microbial food safety, some statistics, data regarding microbial food safety, then microbial hazards of concern, which are the important pathogens which are of concern to human being in different parts of the world, how we get the exposure to these contaminants, and how to protect in entire food chain, and different risk management options available with the government. We have been listening since yesterday on the food safety challenges like increasing population and demand, diversity of food, increasing global trade, and all those kinds of things. One important point which is going to affect or going to change the situation in microbial food safety is changing climatic conditions. It is going to affect a lot of animal pathogens and zoonotic pathogens which come into food chain because there is a change in epidemiological pattern of these diseases. One very good study has been done on Vibrio cholerae in recent years in 2007 in Indian Journal of Medical Research, wherein they have surveyed the Vibrio cholerae affections or cholera in New Delhi area. And it was found that whenever warmer months are shifted earlier, like immediately after winter seasons, the temperature rises. And if there is an early rise in temperatures in end of March or April, there will be shifting of the occurrence of these diseases. Then another challenge we are going to face is the increasing microbial resistance due to indiscriminate use of antimicrobials. Now, this is a workshop focus, and our session will be on microbial hazards. As you know, it needs no introduction. These headlines in newspapers across the world, they are very clear and they tell the story loudly and clearly. Sometimes these news headlines, they are very scary, like deadly spinach. We have been told by Bobby about killer cucumbers. Then we have salmonella outbreaks in different kinds of foods, and so on and so forth. In India also, we have several outbreak reports, and mostly these are confined to large catering events. These are reported from the large catering events, although there are several outbreaks, several cases which are solitary in nature, reported within a family, individual cases which are not reported by these newspapers. But big catering events, they are often get notice of the media. There are several religious parties, marriage ceremonies, and all the kinds of big events which often lead to food poisoning due to microbial pathogens. We do not know exact magnitude of these microbial pathogens causing illnesses, but WHO has estimated that about 2.2 million people in developing countries, they die due to food and waterborne diseases, diarrheal diseases, which are usually food and waterborne diseases. In India also, we have a large number of children having diarrhea. Particularly about 4,000 deaths are reported in children under the 5 years of age, and almost 20 million children, they are affected by the diarrheal cases. There are countries where systematic surveillance systems are in place and estimates are quite reasonably accurate. And like in USA, it has been estimated that about 48 million illnesses, they occur every year with 3,000 deaths annually, and a whopping cost of about $152 billion is there due to these foodborne pathogens. India, we have recently started integrated disease surveillance program in 2009 June, and in which there are several diseases that are covered under this active surveillance program, and food poisoning is one of them. But and you can see from these figures that there is a sharp increase in the number of reported food poisoning cases from 2010 onwards, and in 2012. Half of the year, we have large number of outbreaks. But only drawback in this particular system is that we do not know etiologies of these agents. They just simply report that outbreak has occurred in this particular part, number of cases, number of deaths, that's all. And a sample has been sent for investigation. We do not know the follow-up of that investigation, which is a pathogen which is more important for this food poisoning. And that is very important. We should know which microorganisms are of concern. In relation to food, there are some good fermentative microbes which are essential for fermented food products. There are bad organisms which are pathogens which are of our concern, food safety concern. And in India, we have a lot of spoilage post-harvest losses due to these spoilage organisms which cause huge losses, economic losses. In type of pathogens which cause foodborne illnesses, we have viruses, bacteria, parasites, fungi. These bacterial pathogens, they are one of the most important, and for decades together, work was concentrated on these bacterial pathogens. Several of them, they are there. We know that Salmonella, typhoidal, non-typhoidal, Listeria, Escherichia coli, and all different kinds of pathogens which have been in the center stage for several decades, and a lot of studies they have been done on these pathogens. With advancement in detection techniques, now these viral pathogens, they are gaining more and more importance, and several countries, developed countries, these pathogens, they are the top rankers in the cause of foodborne illnesses. Like norovirus, which is a very important pathogen causing largest number of foodborne illnesses in USA. We have many cases of rotavirus in under 5 age children, and about 1 lakh deaths they are reported every year in children due to this rotavirus. But its association with food or water is still that link has to be established. And then hepatitis A and hepatitis E, both waterborne diseases, we have several outbreaks in India in past with these viruses. One of the most neglected group in foodborne pathogens is the parasites. Although they cause many diseases in India, also we have parasitemia, parasitic infestation in many human beings, but it is most neglected area. We know there are several roundworms, that is nematodal parasites, then flatworms and tapeworms, which cause disease. We have been knowing that trichinella and these measly pork and measly beef, they are associated with food, but due to systematic post-mortem inspection and elimination of the infected animals, they have been controlled largely. But other parasites which are of fecal origin, fecal oral route origin, they are still very important, and Cryptosporidium, which is a protozoan parasite, which is now emerging as an important parasite all over the world. Among all these lists, we should know that which pathogen is important for us or as a consumer, and it varies from country to country. Even within a country, it will vary from region to region. In USA, I have told you that viral parasites, they are by far the largest group of viral pathogens are the largest group of pathogens causing infections. In some other countries like UK and Netherlands, bacterial pathogen infections exceed virus, whereas in Japan, we have almost similar kind of prevalence rates for viral bacterial pathogens and parasitic infestations. they are less studied and less reported. Among different etiological agents, in USA, estimates are there for different pathogens, and noroviruses, as I've told, they are largest contributors in foodborne infections, followed by Campylobacter, Salmonella, and this Cryptosporidium is one of the emerging infection all over the world. Regarding severity of diseases in USA, again, we have a data that Salmonella causes the largest number of hospitalization, followed by Shiga toxin producing E. coli and Campylobacter. Listeria, although cases are less, but severity of disease is more, and there are a large number of hospitalizations, and among viruses, norovirus is having largest number of hospitalization. Situation in India, we do not know. As I have told you that relation with food poisoning and etiology is not yet established, but there are quite a good number of reports on diarrheal diseases in children and in human being adults. And I have taken these figures from the National Institute of Cholera and Enteric Disease annual report. They are doing surveillance for past few years on the diarrheal etiological agents. And this is 1 year figure, but these figures are almost similar for past 3 or 4 years. And you can see here that E. coli group, different kinds of E. coli, they are by far the largest group of pathogens causing largest number of diarrheal diseases, either in a total or in sole agent as a diarrheal agents. Second will be Campylobacter. Among viruses, rotaviruses, they are most important. And in case of parasites, giardiasis or amoebiasis, they are important pathogens for diarrhea. We should know that which food act as a vehicle for these pathogens. We should know the pathogen commodity combination which is most important, and world over there have been identified pathogen commodity combinations like Campylobacter is most often associated with chicken, Salmonella with chicken, vegetable, E. coli O157 with beef, Listeria monocytogenes with cheese, smoked fish and cooked meat. We in India know that Staph aureus intoxications, they are often associated with unhygienic handling of sweets and improper storage, thereby causing intoxications. Clostridium perfringens has an affinity for meat. It requires all the essential nutrients which are provided by meat for its growth. And viruses mostly with fresh produce as well as shellfishes. So in Indian context, although we have scattered information about some association, but systematic studies and systematic data collection analysis has not been done, and we need to identify these important pathogen commodity combinations. How do we get infection? Human, animals, birds, they act as a reservoir. The foods get contaminated, and when there is a proper dose response, enough dose is there and host response is not adequate, then we will have illness. And this dose response, it depends on two major factors, that is a microbial factor and another is a consumer factor. Among microbial factors, there are several microbial factors which are responsible for this risk to consumers, like invasiveness of pathogens, Salmonella, Listeria, they are invading into the cells causing damage. Then pathogenicity, ability of pathogen to cause disease, several virulence factors are there. Within a same species, strains vary in their virulence. In E. coli, Shiga toxin producing E. coli are more virulent than other E. coli. We have to have infectious dose in the food. Some pathogens, they are more stable outside the system or in the environment, like viruses. We have mostly the control measures which are targeted to bacterial pathogens, but we miss the viral agents which are very stable outside the environment, in the outside environment, in the free state. Host specificity. is another important. There are pathogens like Salmonella typhi, Paratyphi A, which are specifically targeting only human beings, and human are the only reservoirs and carriers, and therefore they play important role in epidemiology. In viruses, RNA viruses particularly, there is a lot of antigenic variation, so continuously strains get evolved and they cause infections, and we do not have good protective mechanisms to overcome this particular strategy of viruses. Like I have told you that there are certain pathogens which are more virulent, like Listeria monocytogenes, although cases, illnesses are less, but death is very high, percentage deaths are very high. Likewise, in Salmonella also, they are hospitalization as well as deaths are more. In Campylobacter, although percentage of illness is very high, but the reported deaths are less. Then there are consumers factors like age, socioeconomic status, disease history, and all pregnancy, etc., nutritional status. These affect the exposure and risk to consumers, and as we know, very young, very old, or pregnant, immunocompromised, deceased, they are more susceptible to the infection. The contamination of foods, there are two major sources. One is a primary contamination and another is a secondary contamination. The primary contamination is at the farm level. The diseased animal or carrier animals which harbor pathogens, they are responsible for occurrence of pathogen in the food. Likewise, in plant also, irrigated water which is contaminated with sewage effluents, they cause this inherent presence of pathogens in them. Secondary contamination is one of the most important contributors for contamination of food, and several ways are there for contamination, and unhygienic practices followed in by different people who are handling food is by far the largest contributor for secondary contamination in the food. How we should protect our consumers? This is a food chain, classical food chain, farm produced, then it goes to factory, then to retail, and then to consumer. So we must have barriers or control measures at all steps. That is a primary production, at processing level, at consumer level. And the strategy for this control is, and to check the hazard in the acceptable level, is to prevent the entry of pathogen into food. If it is not possible, then if there is a contamination, try to eliminate it. Again, that step is not possible, then reduce it to acceptable level. After reduction, avoid unacceptable growth or survival of pathogen to the unacceptable level, and then prevent further contamination or spread of pathogens. At primary production level, we have already guidelines available for good agricultural practices as well as good animal husbandry practices for animal and plant foods. And they are very good measures for controlling or preventing pathogen entry into the foods. At processing and consumer level, several approaches, good hygiene practices, good manufacturing practices, HACCP plans and all those things, they can be utilized. At the food formulation and composition level, raw material, raw food ingredients, we should check that they do not have pathogens, and we should always be careful about the food formulation and composition. In the processing step, we can have inactivation steps for controlling these pathogens. We can eliminate the steps which may cause growth opportunities for the pathogens. In packaging, cross-contaminations possibilities can be avoided. During distribution, controls in temperatures can prevent the growth possibility. And at the consumer level, the temperature abuse again can control the possibilities of growth. Different temperature zones which are favorable to pathogens and which are detrimental to pathogens, they are depicted here. As we go in the lower temperatures, although we can stop the growth of pathogens, but we cannot allow, they just remain there and they survive. In higher temperature, as you go up the ladder, there is destruction of pathogens, and at the highest temperature, we can even kill the spores of Clostridium species. We can have a pH control also for the pathogen control, and mostly optimum pH is between 6 to 8. Most of the pathogens, they thrive well and grow well in that pH range. Above and below, we can eliminate these pathogens. For prevention of pathogen in different types of food, we need to have different kinds of approaches which we can use, like 12D concept can be used for vegetables, meat products, reduction in water activity, pH reduction, addition of salts and nitrates can be done, and refrigerations. So these are different approaches which can be options which are available for control of pathogens. Now, government also has options available for policy making and responses for unsafe food. Like yesterday, we were discussing about the prohibition of marketing of the foods and recall of the foods. We can have a mandatory enforcement of microbiological criteria, then process criteria in the operations, import certifications, and we can set up the food safety objectives. Microbiological criteria, there are 3 different kinds of microbiological criteria. One is a standard, which is a regulatory, which is a part of law, mandatory enforceable criteria. Then there are guidelines which are advisory of nature, different kinds of regulators, industries and trade associations and different associations. They have these guidelines which are usually practiced voluntarily. Then we have specifications which are usually mandatory in supplier-buyer relationship, and they are often utilized as control measures. These micro-criteria, they can be at the primary production level for raw material. We can have them at the end product in the processing plant or at the retail, which are usually micro-criteria at regulatory standards which are in the different countries. So here are some examples of micro-criteria which are there in different stages, like end products, retail. Some they have not specified at which stage these micro-criteria are applicable. These criteria, they can be implemented very well with the sampling plan which has been designed and given by ICMSF. And this plan has taken care of degree of health hazard to human being, like we have a moderate hazards on the left side, serious hazard and severe hazard. Then it has taken care of the food handling practices, whether there is a condition which will reduce the degree of hazard, or conditions which will cause no change in hazard, or conditions which may increase. So depending on these parameters, the sampling plan can become more stringent. This is an example of microbial sampling, this microbiological criteria with a sampling plan for cereal-based infant foods. We can have a number of samples, that is N, the number of samples which should comply to the standards, and M is a small m is the number of organisms like Listeria monocytogenes. In 25 grams, 5 samples. Not a single sample should contain any pathogen, whereas in case of like Bacillus cereus, out of 5 samples, 1 sample can have pathogens in the range of 10 to the power 2 to 10 to the power 3, but others, they should not have any pathogen. So this is the sampling plan. We have Indian standards also, but they are just mentioned. There is no associated sampling plan with these regulatory standards. These are examples of the meat, frozen meat, and some other meat products. Then WHO has suggested some risk management options. We have been hearing since yesterday that it should be risk-based, sustainable, integrated food safety system, science-based measures along the entire food chain, which will prevent the pathogens to unacceptable level, and which will be. Then we should communicate the risk, take cooperation from all the stakeholders, and for which this risk analysis based approach is one of the most important, that is to identify and characterize food safety risk with different steps. We can minimize and manage these microbial risk in food production chain, risk management at different level, farm, transportation, and all different levels, and educate, create awareness among all stakeholders with risk communication, different kinds of educational program, mass media campaigns, and all those things. Thank you very much.